For a decade, we ran experiments in the prototype Biosphere, which was a simulation of the pre-flood world. We do know certain things about the world before the flood. We have a handle on the electromagnetic field, which is decreasing exponentially. Every 1,400 years, we lose half the energy in that field. It was 0.5 Gauss. It is today 0.5 Gauss of energy. It was 10 times that because our calculations go back over the chronology of the decades and the millennia. So optimally, it was 5 Gauss of energy. That's what we ran in the prototype, and that's what we will be running in the new large Biosphere. 5 Gauss of energy. That's optimal. Today, it's 0.5 Gauss. Twice I've lectured at NASA on this. Dr. Mario Acuna, the world's leading expert on Earth's magnetic field, agreed with our calculations, and he commended us for the work here. So we have a handle on the electromagnetic field. We have a handle on what the oxygen ratio was. We have a handle on what the atmospheric pressure was. Today, the atmospheric pressure is 14.7 pounds per square inch at sea level. We have calculations. I will not run those past you today. That would take a lot of time. Calculations to show that before the flood, it was about 24 psi, pounds per square inch. It is 14.7. You add just under 10 additional pounds per square inch. You wouldn't be able to feel the difference, but your body would know the difference. And significantly, at 22 pounds per square inch, the available oxygen begins to assimilate in the blood plasma, so that by 24 pounds per square inch, the entire blood plasma is saturated with oxygen. That makes an incredible difference. My wife's life was saved at Texas A&M University, where they put her under 2 atmospheres of pressure. We're going slightly less than 2 atmospheres. It assimilated the oxygen into the bloodstream so that her body was detoxified. She had 22 treatments ultimately that saved her life, and I'm so very, very grateful for that. That also solves the problem of the dinosaurs. The first dinosaur that I discovered, I found to have a small rib cage, the Acrocanthosaurus, discovered along the banks of the Biloxi River. And I was astounded because I hadn't read anything on that, and it was a trade secret of the paleontologist that there was a problem that current atmospheric conditions could not solve. That is, there could not be enough oxygen fed to the deep cell tissue of the dinosaur's body with such a small rib cage, small lung capacity. It would require the creation model. So anyhow, this solves the problem of the dinosaurs. So we have a handle on that. So when we put snakes in there, we put a Grotilus and a Echidrodon contortrix. Those are rattlesnakes and copperheads. So we put rattlesnakes and copperheads in the Biosphere. And we had Dr. Robert Popplewell from Weatherford, PhD specialist, herpetologist, provide the stock and the litter mates to them. And after about 3 weeks, Professor M.E. Clark, University of Illinois, tenured professor, came down for his 38th trip from the main campus to visit Creation Evidence Museum. And M.E. said, why don't you milk the snakes? I said, M.E., to the professor, I've said a number of times that it would take the third generation before we'd be able to see any difference in the venom. And he said, well, you've been wrong before. Thanks, Prof. He said, there might be some difference already. I said, it hasn't been long enough. He said, it won't hurt, so let's milk them. So I said, okay, M.E., you milk them. No, I don't touch snakes. You milk them. I don't touch snakes. No. I had a specialist put them in. So we brought Robert Popplewell and Robert Summers. They play with snakes. I mean, they love it. We have the documentation of the milking the snakes, and we put them under freezing conditions, etc., the venom. And then M.E. Clark and I transported them to an independent lab in Dallas. They ran them through scanning electron analysis, and to their absolute surprise, on one side we have the copperhead litter mates. Same litter, 40 miles away, brought them in, milked them. It's all gnarled like spaghetti. But after just 3 weeks in the biosphere, the complete restructuring had occurred. The same thing with the rattlesnakes. And this is your copy at the back. I won't read this. Same thing with the rattlesnakes. And Dr. Bowles said, you have to let me publish on this. On the back is a letter that he sent to me, a copy of a letter. We let him publish on this, and he published in the technical literature stating that we now have a new avenue to completely alter the basic matrix structure of venomous creatures to chaos to structure. And this opens new fields of investigation for scientific research in epidemiology and other areas particularly that have to do with the environment affecting epigenetics, the environment affecting genetic expression. I'll state one other thing that's not on the list. He then said, let me put 2 PhDs on this and we will do electrophoresis. So we did. Took our samples, ran them through electrophoresis, that is, the Western blot test. Some of you with a medical background understand that. The Western blot test runs DC current through the protein, and you get 4 columns of expression of the signals of the DNA, of the protein structure. And he called me to the lab and he said, this has never been done before. He said, this is a column showing where, compared to the litter mates, you have increased the strength of that genetic expression. Here, you have diminished that expression. Here, you have eliminated that expression. But here, you've expressed proteins that are not expressed in the litter mates. You've actually performed a different expression that destroys evolutionary theory. Changes can occur in biological systems. Traits can occur in biological systems not because of evolutionary development and mutation damage, but because the information is already in the original organism. Do you understand what I just said? Charles Darwin, it's time for you to sit down. Because scientific work going on at the Creation Evidence Museum offers an opportunity for a scientific explanation for the variation we find throughout God's creation.